Um, can you hear me? Great. No, I think it's on. Do you want me to hold it? No? Is it on? I see people nodding. Okay, great. Because I find it really difficult to stand still. So, so I'm going to walk around and try not to also hide my slides while I talk. And first of all, what I'd like to do is to express my excitement about being here. I've ever since reading about ECODIM, about knowing about this course, I've wanted to be part of it. I had hoped in 2018 to join, but gave an online talk that I think was pretty scrambled for technical reasons. And so I'm super excited to be here. Thank you for the invitation. And I'm very excited about spending the next 2 weeks with you in this course. And of course, probably all of you know Pablo Neruda, and I don't know if you've read his poem or ode about the sea, but it's deeply moving and impressive. And the statement that he needed the sea or needed the sea because it teaches him is very relevant to the discovery of hydrothermal vents because nobody knew about them before they were discovered. They were discovered just off the Galapagos Islands. And I like to emphasize that they were discovered only 45 years ago. And of course, many of you in this room were not even born then or a twinkling in your parents' eyes. But the reason that is interesting, there are two. The researchers that discovered them and that understood what drives them, many of them are still alive today. And I really, really encourage you, if you get a chance, to hear them speak. It's very, very exciting researchers that were at the forefront of a field when it happened. Another reason is I claim this is the last major discovery of an unknown ecosystem on Earth. I would argue that that's it, that we've discovered everything, including in the deep sea, that there is in terms of large communities that we knew nothing about. And so their hydrothermal vents support tremendous biomass, and it's huge per square meter, as large as that of tropical rainforests. And the primary production, as you know, there are only two forms of primary production on Earth. One is through light, photosynthesis, I'll explain that in a second, and the other through chemosynthesis. And what is so fascinating, in the deep sea, there's simply not enough organic material for animals to live on, at least to build these kinds of large communities. And so these communities are fueled entirely by symbioses with chemosynthetic bacteria. And so very briefly, many of you are familiar with chemosynthesis, but particularly for the students that might need to continuously think about it again and again, is in photosynthesis, you've got the energy from the sun, that the chloroplasts, which of course are descendants of symbionts, cyanobacteria, that are fixing CO2 into biomass for the host. In chemosynthesis, the energy, we like to call it dark energy because it's not from the sun. The electron donor there are reduced compounds like hydrogen sulfide, like methane, like hydrogen, and they're taken up by the symbiotic bacteria. They use those electron donors. They have to oxidize them. They need an oxidant, so they need oxygen, and the animals do as well, to fix CO2, and then that biomass is transferred to the host. Although that's an elegant way of putting it, the host actually eats the symbiont. So they do all that work for the host, and then their reward is that they're eaten. What is interesting is we had to go to the bottom of the sea in 3,000 meters water depth to discover these symbioses. And very soon afterwards, it was discovered that they're not only at hot vents, but also at cold seeps. And whale falls, when they sink to the bottom, very cool, their bones degrade. They're full of oil and sulfate reduce that to sulfide, and they have communities there. But what is truly remarkable is that these chemosynthetic symbioses are widespread in shallow water environments, and those are much easier to access than deep sea environments. And for literally 100 years, zoologists were often surprised to find shallow water animals that had a reduced digestive system, but they assumed that they were taking up organic material from the environment. And it was only after the discovery of the symbioses at the hydrothermal vents that the zoologists went back and looked at these animals, actually looked at them with electron microscopy and said, oh my God, they're full of bacteria, and these bacteria could also be using sulfide. So it's a lesson that we learn continuously in the sciences, is that we have to learn something to then understand it fully and interpret it and see what we're doing. And I like to really emphasize that, particularly to students, because you're going to be discovering stuff that we don't know now, and where in 20 years from now we're going to go like, how come you didn't know that, or how come you didn't see that? So this is a really good example of that. What we now know since these 45 years of discovering these symbioses, is that they have evolved numerous times independently of each other in convergent evolution. And so what you see here is a phylogenetic tree of bacteria that are associated with different animal host groups. And we now know that at least 15 bacterial lineages, from the gamma proteobacteria, from the alpha proteobacteria, from the what used to be called epsilon proteobacteria and is now called Campylobacter. I hate this whole renaming. It's really a struggle. But you can see that there are many different lineages that have established this in parallel independently. And similarly, on the host side, with both 2 protist groups and 9 animal groups, and I'm sure we can discover more, for example, here in regions of the world where we have not sampled very well, that we will continue to discover new associations. And what that tells us, when we see something happening again and again in convergent evolution, is that there's a strong selective advantage, both on the side of the bacteria and on the side of the host, in associating and coming together, particularly in environments where there's not enough organic material for the animals to survive. These are some of the hosts that we work on in my lab, and I'll in the next slide, I'll show you a little bit more about gutless oligochaetes. Oligochaetes are like earthworms, same group, annelids, and they're very, very small worms that occur in shallow water environments that we worked on a lot. Nematodes that have endosymbionts, ectosymbionts, different group, flatworms, Polycotinula, ciliates, deep sea mussels that I'll spend most of my talk talking about. And then this is actually cheating. This is an animal called a placozoan from a very, very early branching lineage of life. It doesn't have chemosynthetic symbionts, but some very cool other symbionts that we've worked on, and that you can find in a nice paper that we worked on here. But these are the groups that we work on. My hope is that in the time that we have here, we might be able to find some gutless oligochaetes here in these environments, wherever there's quite a bit, or actually it doesn't take that much hydrogen sulfide and enough oxygen. These are environments where we can find them. And so I'm hoping, particularly with Thioploca and Thiomargarita being around here, that we might have a chance to see them. This is a gigantic blown up images. They're very small. They're as small as like a little piece of white thread, but they're very easy to find. And they've been found off in the Peru margin where they're extremely abundant. So they should be around here. All right, so in our research, we take an approach where we like to say from genes to the environment and back again. I would say that we describe ourselves as organismal biologists in that we really, really try to understand all aspects of the symbiosis and verify particularly hypotheses that we've developed through the omics, then through experimental analyses. And I'll show you some of that work then in the next slides on the deep sea mussels. To do that kind of work, we need an extensive toolbox and a lot of funding, which I'm very fortunate to have received. So we do in situ experiments and collection work, lots of different imaging methods, incubations, and the omics. So for the rest of my talk, I'll be focusing only on the deep sea mussels that we work on, and hopefully over the next 2 weeks, I'll have time to tell you more about all the other host groups that we work on. So as all chemosynthetic symbionts so far, nobody's been able to cultivate them, which is not so easy to understand. Many have a free living form. They should be cultivable. Their genomes don't reveal anything that would make them very difficult to cultivate. But then again, when you think about the paper that came out on the Loki archaeota, where it took the Japanese 12 years to culture them, I think, and they invested a lot of energy in it. Nobody in my lab has invested that kind of energy. I always say to students or to postdocs, sure, you can give it a try, but after 3 months, you have to move on to another project because otherwise you're not going to have a paper to publish to come out. But what we can do is we can keep the hosts in the aquaria and we can keep them there for many years, and that means we can manipulate them. So there's a lot you can do if you can keep the host. You don't have to always cultivate the organisms we work on. I know I'm preaching to the converted, but there are a lot of people in the host microbe world that feel if you can't genetically manipulate the partners, and it's really not worthwhile working on. I'm going to be describing some of the imaging work that we've done, and you can see that we have a very biased team here of only one gender, but it does get better as when I show you some of our other work. So Nicole is now at EMBL, will be heading one part of the trek of the Terra Oceans Transversing European Coastlines expedition. Benedict Geyer, who worked on the imaging during his PhD with me, now as a postdoc at Stanford, and Max Franke is now at Zeiss Microscopy. What you see here is an electron microscope image, a tomography image, micro CT, microcomputed tomography of a muscle that has been opened up, right? So this is, if it's you open it up along its hinge, this is the foot. And here you see these, in real life, big, thick, fleshy brown tissues. These are the gills. The gills are extremely enlarged. We've highlighted them here in yellow, much larger, I'll show you in the next slides, than they are in non-symbiotic mussels. And the reason that they're so enlarged is because they're engorged with the symbiotic bacteria. And what these are low diversity, high abundance bioreactors, because each single mussel, a mussel of about 8 centimeters length, which is pretty small, can have up to 10 to the 12 bacterial cells per host individual, which means that when we're doing the omic analyses, it's a lot easier for us than, for example, poor Nicole having to work on sponges where the abundance is much lower and the diversity is much higher. And as a rule, there are only 2 bacterial species. It's actually a little bit more complicated, and I can talk about that another time. They do have a parasite, but the dominant species are a sulfur oxidizer and a methane oxidizer. And they live inside. This is now a live mussel. These are the brown, thick, fleshy gill tissues, and the symbionts live inside the gill cells. imagine, if you will, just so that you can understand where they sit. It's actually something that a lot of people, not only students, professors, scientists, struggle with. Imagine a radiator, and these each of these elements are a filament of the gill, right? And so the outside here, let me show you. This slat here, this filament, is comparable to one of these slats. There's seawater streaming around the outside of these, just like there's air in a radiator that is streaming past the slats. And inside is the muscle tissue, the muscle fluid, sorry. It has a circulating system with hemocytes, so like the blood system would be. So just like inside a radiator, there's the hot water. Then you get an idea of how they're built. And now imagine if you wanted to combine a radiator with energy from the sun, and you could put phototrophic bacteria in here, then you would actually have, this is the first time I'm developing this model to explain it, but I just thought that's a really, really cool way of providing energy. Forget it. Sorry. Okay. So what I would like to show here, though, is that this is the outer edge of the gills that are directly exposed to the seawater. There are no symbiont cells in here. We use fluorescent in situ hybridization with probes that are specific to the sulfur oxidizer and the methane oxidizer. They are only on the inside parts. These cells that are symbiont-free and those that contain symbionts are immediately adjacent next to each other. So there's a clear ability of the host to say, you can colonize these cells, but you can't colonize other cells. How they do that, we don't know. We need to do single cell transcriptomics or proteomics to do that. And so if you zoom in a little bit more, we're now looking at a single host cell, a single bacteriocyte, and we see the sulfur oxidizers on the outside towards the seawater. The methane oxidizers are more towards the basal side of the cell, whether that's about availability of electron donors or what's driving that separation, we don't really know. And if you look in a little bit more closely using electron microscopy, again, the sulfur oxidizers here and the methane oxidizers below. All right. So the gill cells are modified to eat the bacteria through phagolysosomal digestion. So it's like a conveyor belt. You have the symbionts here. They're dividing and growing, and over time, as they are phagocytized as they move down towards the basal part of the cell. There's a second way that they might also gain their nutrition from them that I don't have time to talk about now, but again, would be happy to talk about later. So the hosts have adapted their body plans to host these microbes, just as ruminants do, as many hosts that house symbionts. What you see here is a shallow water blue mussel, and these are the gills, and you can see they're very flat and shallow. They're gill cells and very skinny looking and skimpy looking compared to these big, thick, fleshy gills that the deep sea symbiotic mussels have. And now you can imagine how much work it is for poor non-symbiotic shallow water bivalves. They have to filter water all the time consistently to get enough out of the water column to eat. And then you can imagine why the biomass of deep sea mussels is about 5 times higher than it is than their shallow water relatives, is because they can kind of sit there and just farm their bacteria and live off of them because there's enough energy in their environment through the sulfide and the methane and the oxygen that is there, the oxidants that they need. So once you've actually, that's the selective advantage I was talking about. Once you've been able to establish a symbiosis like that, you have an incredible advantage. Another adaptation is that a normal epithelial cell, also in your gut tissues or any animal epithelial cell, has microvilli at the apical, at the top end of its cell. And what happens to the cells that take up the bacteriocytes, that take up the symbionts, they go from a normal looking cell to one that loses its microvilli. And that's an intriguing phenomena because we know that pathogens also induce the loss of microvilli. And whether that's closing the door behind them, this is what is assumed with pathogens, so that the pathogen needs these microvilli to enter the cell, or what the mechanisms are behind that is something we're still trying to understand. Also interesting is the simplification of the gut. So as I described with shallow water mussels, it's hard work to eat plants and algae, and that's why ruminants have such an extended gut. It's because you really need a lot of space for your microorganisms to digest it. And similarly, when you look here in red at the intestine of a shallow water bivalve, it's looped to extend it so that you've got enough time to eat it. When Max Franke did his PhD on looking at the development of the early stages of the deep sea mussel, you see that when they're larvae, they still have this looped intestine, which is actually very cool because they might be going to the surface and feeding on phytoplankton and going back down. That's a whole nother story. But then when they settle on the seafloor and have an easy life with eating with living in symbiosis with the bacteria, their intestine goes from this complicated loop system to a straight gut. So a much simpler gut system. Somebody here sitting in the audience, I believe it was you, was asking about symbiont transmission. And so we differentiate generally between horizontally transmitted symbionts that are taken up from the environment and then vertically transmitted symbionts that are passed on from one generation to the next. It's never that strict, though. That's important to understand. There are always all sorts of exceptions. But what we do know here for the deep sea mussels is that they take them up from the environment. And we were very lucky during a research cruise to be able to collect these stages. We had never collected them before. And again, it was a French zoologist that works on very, very small larval stages that joined us during a cruise. And we had said during numerous cruises, we can't find them, they're not there. And he looked and he showed us what we were overseeing as little sand pebbles, which were actually these larval stages. So that was a great advance for us. And we've been able to look at them. We do think that they go all the way up to the phototrophic zone when they, in their early life stages, to feed, and then drop back down. How they find the vents and seeps, no one knows. Maybe they don't find them. Maybe there's just enough progeny to then whoever misses it, doesn't succeed. That's not clear. And so what we see is in the gill cells of the juvenile larvae stages that are settling on the seafloor at the vents and seeps, that they begin to take up the symbionts. And so they're only taking up their symbionts from where they settle, right? If they don't settle where there's energy sources, they're not going to be able to take up symbionts, and they might have to try to filter feed and get by on that little bit. That's not going to be an easy life. And they enter at the gill surface, which you can see here with the methanotrophs and the sulfur oxidizer. And intriguingly, the sulfur oxidizer always colonizes first, and then the methane oxidizer. But there are some mussels that have only methane oxidizers, so that works without the sulfur oxidizer. Okay, and here you can see this ruffling that is known in host microbe interactions also, and in pathogenic associations. As I said, we can keep them in aquaria, and we collaborate with our colleagues in Kiel at Geomar. We've collected mussels from the Gulf of Mexico, and then we kept them in aquaria. And we kept them, in the beginning, we were super nervous about having methane with oxygen in these closed in a temperature controlled small room, because any spark is going to possibly cause quite a fire. So we were super careful in the beginning and only had but constant flow through 10 micromolar methane. And the reason we ramped it up to 40 and now we're at 80 is the following. This is what this is to scale. They're both at the same size, same resolution. This is what the bacteria site looks like when it's freshly collected, full of symbiotic bacteria. And these poor mussels at 1 year at 10 micromolar methane constant flux they completely are starved. They don't have their symbionts anymore. They've lost them. So that's not enough methane for them, which is surprising in terms of how we think about if a flux is constant, concentrations can be low. We ramped it back up to 40, and you can see that they've taken up more. At 80, we're just looking at that micromolars methane, whether it will increase. How high we need to go is something we're still looking at. That's it for the imaging, and I'll move on now to the sequencing that we do. Have you timed me? How many more minutes do I have? I've still got 25, but with the questions, right? Yeah, okay, great. So I'll try to go through the sequencing in about 10 minutes, 12, so that you have time for questions. So I'll move, this is our gender biased other team that does the omics in our lab. And Jill Peterson, who now has her own lab, or now actually for over 5 years now, at the University of Vienna working on shallow water leucinids. Rebecca Anzaga and Liz Zayavedra, who are now at the Quadram Institute looking at the human gut microbiome. And Male Oecke, who joined a biotech company in southern Germany. And this is something that is very dear to my heart and that I'm happy to talk about also during the next 2 weeks. Even in this day and age where we have so many methods, omic methods for sequencing, it's extremely challenging to identify strains. But I'll come to it. All the action is at the strain level, in my opinion. I think that our focus on 16S means that we're not actually looking at where the different environmental niches are in the environment. You can try to cultivate microbes, and Martin Pols and others have done great work with Vibrios, but you literally need to run from the environment back to the lab and sequence it, because use it or lose it, and you're growing them on a certain environment that is going to bias what you're going to find in the genome. Bacteria are extremely efficient and fast at losing genes and at gaining them. And so it's really been, I really can't emphasize it enough to students, but this is the golden age of working on environmental microbes, because now we have the tools with omics to really see what's going on. It's super exciting to be part of this. And of course, with long read sequencing, I think Nature just declared long read sequencing to the method of the year. It's incredible what we can resolve at the strain level. The single cell amplified genome, something I'm happy to discuss, is not working so well so far. So I'm more convinced that metagenomics, long read sequencing is, at least for us right now, that's working better. But the point I'd like to make is that when we look at 16S sequencing and with ASV, with variant sequencing, if you can distinguish, let's say, 1% difference, the time that it takes to evolve 1% difference can be as much as 50 million years. Right? So that has nothing to do often with the evolutionary rate of the hosts that you're looking at. So it's something you need to think about. You're looking at like you're comparing humans with fish and snails. That's how high up you're looking down at the species diversity versus this is where a lot is happening. All right. And of course, the reason I think that is because that's what we see in our symbioses. But honestly, it's also in a lot of free living communities. It's the case. And I'll show you what we found. We looked at 4 closely related mussel species that are on hydrothermal vents on the Mid-Atlantic Ridge. Naming these mussels is a challenge because there you have to go back to morphology, just like it was in microbiology, where you needed to cultivate them to say them. Same here. Somebody has to look at them morphologically. Those people are dying out and no longer getting, well, they didn't get jobs. So now we have these creative names like Bathymypdilus species 5 degrees south, 9 degrees south. So that's something that also in the animal world is going to have to be resolved. And you can see at 16S, each of these different species has a specific symbiont at the 16S level. But again, emphasizing, even if we're off here by a factor of 10,000 years, these are long divergence times that you're looking at for 16S. So what Rebecca did in her PhD was dive down into looking at the strain diversity, and this is published a couple of years ago, so I won't go into the methods, but happy to talk about it. A single mussel individual, which has a single 16S phylotype of sulfur oxidizing symbionts, can have as many as 16 different strains of this phylotype. So a high diversity at the strain level. And when we first saw these results, we got a lot of pushback from evolutionary biologists because what they said is in very intimate intracellular symbioses, the selective pressure is against that kind of strain diversity because they're competing for resources within a cell and they're also competing for space. So how can we explain this kind of diversity that we're seeing? And also a great advantage for the non-cultivable world is that it's finally relevant to humans because we're so interested in our own microbiome. And that's been giving us a lot of flow back and forth in terms of the methods they're developing, our insights and their insights. And so what we do know from many different host microbe analyses is that the composition of symbiotic communities can be shaped by the physical location, host genetics, and environment. And so one of the things we're interested in is, could it be that each single bacteriocyte hosts only a single strain? So maybe only 1 or 2 symbionts get in, and they close the door, and that's like, now we're going to multiply in this cell. And there's a beautiful parallel with colonization of your skin pores in your face, where a fantastic researcher at MIT, Tammy Lieberman, has shown that only a single strain colonizes each of your pores and then kind of closes the doors for the others that follow. So that's one possibility, and we're trying to figure it out. It's extremely challenging to get these cells to separate, but that's another story. What we do know, I'll go through this quickly because it's also published, is host genetics does not appear to play a role in shaping the microbiome. We're extremely lucky to have a hybrid zone where we have the 2 species, the host species mixing. And so that gave us a beautiful setting to say, well, are there differences between the 2 host species and their hybrids, or do they all have the same symbionts and the same strains? And what we see here, these are the hybrids in green, and these are in yellow, the parental generations, is that sequence comparing 171 single copy marker genes, doing entire metagenomes, reconstructing them. We don't see differences. And we also looked at presence, absence of genes and SNP diversity and all sorts of other stuff that you can do to try to see if there are differences. So what we think is shaping it, and this is the same for free living microorganisms, is the environment itself. That is driving the strain diversity and the selection. And the reason that we think that is Rebecca very nicely showed that the strains vary in their functional repertoire. And what you see here is the consensus. This is read mapping from Illumina sequencing. This is the consensus sequence of a sulfur oxidizer from a single mussel up here. If all of the strains have a given gene, like let's say the ribosomal genes, because they're really important, then when you map the reads of your sequencing from a single muscle or from the environment, then you should see those genes mapping 100%. In other words, they're present all the time in all the strains that are there. If only some of the strains and not others have it, then you won't see that kind of high read coverage. You'll see a much lower read coverage. These are the analyses, the type of analysis that Rebecca used to identify strain-specific genes. Very nice method that she developed and used for this. 80% of the genes couldn't be annotated. Nicole showed that too in her talk. That's standard. We're used to that, but we super lucked out with the 20% that we could annotate because a lot of them make sense in terms of selective pressure. Virus defense genes, CRISPR-Cas, toxin-related genes, and metabolic genes. And so for the last few slides, I'll focus only on one advantage of having a gene that will drive strain specificity. I should emphasize, by the way, that these genes are expressed. We find them in the transcriptomes and proteomes, so we're not talking about some pseudogenes. And also, it's intriguing, many of them are acquired through horizontal gene transfer. When you do phylogenese, you can see that. All right, so just summary, everything nitrate, phosphate, phage defense, lots of very cool stuff that we'll probably need another 50 years to really look at fully. But let me just tell you about hydrogen. So we know that the sulfur oxidizers, we showed that and published that quite a while ago with Jill as the first author, that the sulfur oxidizers can use hydrogen as an energy source in addition to sulfide. The problem is some vents have a lot of hydrogen, but some vents only have very little hydrogen. It's not available at all vents for geological processes. It's called a certain type of rock setting that gives you a lot of hydrogen. And what Rebecca saw is these are the 4 different vent sites that you see here. So what we see here is Clueless, this is Lilliput, this is Semenoff, and this is Lucky Strike. What we see here are the percent of the sulfur oxidizers that have the gene, the hydrogenase gene that allows them to use hydrogen as an energy source. And here at 2 of the vent sites that have the highest hydrogen concentration, 100% of the symbionts have the genes that allow them to use that. In fact, they have the whole operon. Down here at Clueless, there are low hydrogen concentrations, higher sulfide concentrations. The proportion of strains of sulfur oxidizers that have the ability to do that is much lower. So it's the environment that is driving that. And we are able, and I get goosebumps because I did this experiment with trying to see if they use hydrogen and incubating it into glass vials at this vent site. And it's really hard to do in physiological experiments. If I had done it at this vent site, because I just thought, why, just test it, it never would have worked, and I never would have followed this story. So that's another serendipity experience, in that you're lucky sometimes with your research. So the environment is driving it. Miguel Gonzalez Porras has done some great works with visualization of the gene itself compared to a 16S gene, to show that there's actually some kind of spatial distribution here in the different symbionts that have it. So wrapping up, the advantages of strain diversity. The muscle can continuously exchange strains with the environment, with co-occurring hosts, but also with the free-living bacteria that it can take up. And it allows the muscles to adapt to the vent environment in which they are. Lots of hydrogen, I'll take symbionts that have the ability to use that energy source. And it's rapid, that adaptation. It's within their lifetime themselves. They don't have to wait until their progeny or their grandchildren evolve the ability to do that. It's immediate. And with that, again, I mentioned the people that did a lot of the work. Here are the funding sources that did this, and in just 2 more slides, We have a fantastic graduate program that I'd like to advertise here, also very often with joint degrees from other countries and other universities. So we're not taking away students from other countries, but rather trying to collaborate with them. And as you can see here, we have really an incredible diversity also of nationalities. And I have to say that the students so far, at least from Latin America that we've had, continue to be some of the best students we had. So if you're interested and your advisor is interested in a collaboration, let me know. And also I'd like to, that got mangled, but it's the only one so far, is that there are some fantastic programs in Germany with where funding can be provided at any level of your training to come and work in Germany. So if you're interested, do let me know. And I'll thank you for your attention, and I'm looking forward to your questions.
Thank you very much for your talk, Nicole. That was wonderful. So we have time for questions. Somebody there?
Okay. Yeah, okay. I'll stick there. Questions?
Hi, Nicole. Thank you for your amazing presentation. I feel curious. So you show us how the gill of the mussel was modified after a year. You have the culture, the mussel in the lab, but it appears that the modification of the gill that you showed us before was reversed. So you also have the small fiber that they have.
Microvilli, yeah.
Mm-hmm. Yeah. So do you think, so when you change the conditions, they recover the same like morphology they previously have with the symbionts in the environment? So do you think that microorganisms are driving those changes in the host?
Yeah. So as I interpret your question, there are lots of different corners I could go to. The first is the loss of the microvilli. That always happens. That doesn't matter whether it's in the environment or in the aquaria. So that part is consistent. It's the concentration of methane that influences how many are taken up. What we're still analyzing is when they regain their symbionts, did they take up the exact same strains that they had in the Gulf of Mexico? Or did the environment in the aquaria change? So that's something we're looking at. The methane oxidizers strain variability is lower, but we're super interested in how that changes over time because an aquarium environment is very different. And your last question, who's in the driver's seat, is one of our favorite ones. Like, is it the host that is determining the symbiont community or the symbionts or both? It's a very basic and fundamental question in host microbe research.
Well, thank you. Can I do another one? Sorry.
Mm-hmm.
Just because you mentioned this, and I was thinking, in the aquarium, you actually will not have the environmental sources of microorganisms. So they stay in the cells and they change just the abundance when they are.
Yeah, thanks for asking that question because I didn't explain it well. Remember when I said that they eat their symbionts through phagolysosomal digestion? And I said, well, but there's another method. Benedict Gaia was doing some amazing work, and I'm doing this because it's a radiation synchrotron facility where the electrons are going through a path of 3 kilometers at Daisy in Hamburg. And it allowed us to actually go through high resolution through the gills. And he said, what are all these blobs that are off the gills? And they're blebbing these bacteriocytes like crazy. They're losing them. And he's now followed them with fish. And what they do is then, this is how a normal mussel feeds, is you have your cilia and you're pushing your food down through a food groove into your mouth, and you're pooping out actually a huge proportion of the bacteria at the end. It's called pseudofeces. That's what you don't eat. Anyway, it's complicated. What he could see is that there are viable symbionts. In other words, they give a fish signal full of ribosomes coming out the back end of the mussels. So our hypothesis is that they're actually, just like farming, that you're also getting a release of the symbionts back into the environment. And we think that that would also happen in the aquaria. So that would explain how you could still have the same strains. Also at vents and seeps, you have predators, crabs. I have videos that I can show when we have more time in the chorus, but it's very cool. You can see the crabs just like crack open the mussels and eat them. That will release the symbionts as well.
Oh, thank you very much.
Is there another question? Okay, sorry.
So hi, wonderful talk. I just have a question involving the bacteriocytes that are in the gill of the mussel. So it consists of, is that there been any research done on like a system of communication between those bacteriocytes, or they just like close the node, any way of interaction between them or something?
Do you mean communication between two neighboring bacteriocytes?
Yeah.
Sorry, I'm really thinking about this. The first question is, why would they want to communicate beyond what any neighboring cell in an animal or a eukaryote would need to communicate? We don't ever see evidence of bacteria moving between cells. So in that sense, a communication laterally would not be important. What we do see is this normal turnover, like a sloughing of the epidermal epithelial cells. But that's standard. That's in your gut too, as well. You're continuously sloughing off cells. Same, by the way. Any epithelial tissues, there's a high turnover rate. But communication between, is there a reason you're asking?
No, I'm just curious.
Oh, that's fine. That's what drives a lot of our questions.
Okay, thank you.
Thank you very much for the amazing presentation. What the mussels gives to the symbionts?
Yeah.
I would like to know for my next question, please.
Yeah, exactly. Yeah, it's an excellent question. What's in it if you're going to be eaten? So what matters for any species is to increase your population size. It's never the survival of the individual, but rather the survival of the population as a whole. And so the hypothesis has been that if you think about a poor, free-living chemoautotrophic bacterium at a hydrothermal vent. The interface between oxidants and reductants is extremely narrow. And then you've also got the currents, and so you have to attach to a surface where you're getting enough diffuse fluids, but also enough oxidants. You have to compete with anybody else that wants to colonize that surface. So the area that you can colonize is extremely small. Now think about these huge mussel beds. You're already colonizing a much, much greater area. So the advantage on the short term is you're getting the oxidants and reductants. The animal is providing them to you. You're increasing your population size. All of that will only work if you get back out again, right? for the population size to increase. And that, as I just explained, happens. But we also have to be careful because there are bacteria that become enslaved that are only vertically transmitted. In insects, we know that. There's no advantage whatsoever. They will eventually die out, and the insect symbionts will take up a new species. So it's not always, there's not always an advantage for the symbiont. But here there is. Here we think there is.
Okay, so that brings my next question. Like, have you ever think about if it's a little bit difficult to take the organisms and to incubate them because you lose all the environmental conditions and other like hidden conditions, why you don't bring like a model mussel, for example, into the vents, so you start you have your model like 3D printed mussel, for example, with the gills, and then you can study the first colonizers and the second colonizers, like you have the model that you can bring it down there. So could be like a, because if the mussel serve as a house for them, you can build them a house and then you can study what is going on in terms of colonization.
Yeah, excellent thoughts. So what we do is we do so-called displacement experiments in the deep sea. So we take the mussels, we move them away from active venting. The cruise is never long enough, that like 14 days is the most, and then actually replacing them and getting good ROV pilots that are able to do that. So we're doing that. We've also developed methods to fix them in the deep sea. So we took RNA later 3,000 meters down and had the pilots crush the mussels and throw them in little RNA later pots so that we could be sure that what we're looking at is what they're doing in the deep sea. I don't think that bringing shallow water non-symbiotic mussels down would give us much information because they won't take up the bacteria. They won't take up symbionts. Most bivalves that are non-symbiotic, pathogens or E. coli, whatever, they all end up in their hepatopancreas. It's very, very unusual for epithelial cells to take up bacteria. So there's already some adaptation that has happened that will not happen in non-symbiotic ones. It's, by the way, again, very similar to our epithelial lining in our gut. It's very, very hard for a pathogen to get into it. A lot of the pathogens are on the surface or on the mucus, but getting into a host cell, that's not easy. So that's why we wouldn't take a non-symbiotic host.
Okay, perfect. Thank you.
More questions?
Thank you for such a lovely talk. I was wondering, with the locations of the symbionts, whether anyone's tried to do a technique like matrix-assisted laser MALDI to try and look at maybe the lipidomic or the lipidome of the cells that have been kind of colonized versus cells that haven't been colonized.
Thanks for that question. That was actually how I got my grant from the Gordon and Betty Moore Foundation, was that I said that I would develop MALDI imaging to look at metabolites, also lipids, in symbiotic associations and increase the resolution. And when I wrote that grant, I was completely clueless about how to do it. So I still am amazed that I got it. But I was extremely fortunate in being able to recruit an excellent scientist who I haven't presented here, Manuel Liebeke, who is doing exactly that. And thanks to also developments with MALDIs and the lasers and the methods they do it, we're down to a resolution of it used to be only about, when did I get the grant? 10 years ago. The resolution with MALDI imaging is about 20 micrometers. So then you're down that's not very relevant. And with the newest instruments developed in parallel, actually for materials research, but they're great for biology, we're down to a few micrometers now. So we're very, very excited. And of course, lipids are fantastic for imaging because they actually stay in the tissues and are not blown off by the ionization. What we are seeing is certainly interesting in that the host microbe world has focused a lot on sugars. It's always been assumed that sugar is also in rhizobial symbioses are what matters, but in pathogenic associations, it's actually lipids. That's an incredibly energy-rich source. And so more and more now, the host microbe world is starting to look at lipids, and we're seeing some very interesting, not so much at the imaging, but at the omics level of what's going on. But it's all so preliminary that I can't tell you much that you might find interesting.
Hi, thanks. I wasn't sure if I to do the question because it's a little bit of a follow-up of the previous questions, talking about the specificity of where the symbionts are and the sulfur oxidizing and the methane oxidizing. And so basically, since he asked the question basically with more information, so we think it's lipids that are determining this specificity, because and these lipids are the ones that and are these lipids the ones that are making this symbiosis specific to the type of bacteria, but not to a specific bacteria, because
So if I understand your question correctly, it's okay, is I think you're asking about the recognition, like how do I know as a host who I'm going to let in and who can't come in? It's like, you have a big party, right? And you don't want it to be crashed. You only want your friends to come in. And so we think that recognition is going through cell surface recognition. You might have seen with the strain variability, there were O antigens that I was showing that were very variable between the strains. So there, we're trying to understand better how that works. The lipids, we think, are unspecific in terms of strain variability and recognition, but rather more important for the nutrition.
Thanks, Nicole. Maybe you written about it already, or the hydrogen oxidation operon. Do you find it in mobile elements?
In German, the combination of yes and no is jein, ja and nein. Yes, we find mobile elements, but they're not directly downstream and upstream of the operon, but rather a few genes separated, which of course over evolutionary time, depending on when they were transferred, would be perfectly reasonable that they would be moved away. They certainly have a phylogeny that is completely indicative of horizontal gene transfer. They're more closely related to, I have to go back and look at it in the tree, but they're not gamma proteobacterial hydrogenases. Then the nucleotide usage patterns or the GC content is a little bit different, but again, that will change over time. So as you acquire genes horizontally, in the beginning, it's really easy to pick them out because they're so different. Yes, we see them, but it would be nicer if they were closer.
Okay, thank you, Nicole. I know that we have some questions, but we have to move to the other talk. So you can talk with Nicole maybe lunchtime or in the afternoon. So thank you, Nicole.